Hi, everyone. My name is Arielle Keller, and I'm a postdoctoral fellow at the University of Pennsylvania. Today, I'm excited to discuss some examples of ways that machine learning can facilitate discoveries of generalizable associations between neuroimaging measures and cognitive and clinical phenotypes. So I want to start by having us consider why it might be important to take a personalized neuroscience approach to some of the major problems that we care about in human brain mapping. The first reason is that there are profound individual differences in cognition that emerge early in development and can become more pronounced with age. Cognitive impairments are associated with a number of adverse outcomes, including lower quality of life, increased risk-taking behaviors in adolescents, and an increased risk for psychopathology. So it's important for us to understand the neural bases for these individual differences. Second, we know that there are substantial individual differences in psychiatric disorders. Not only is there profound heterogeneity in presentation within diagnostic categories, there are also similarities across diagnostic categories, implying the need for machine learning models that can parse underlying dimensions of inter-individual variation. Third, not all brains are alike. So while we know that there's plenty of structural variation in brain morphology across individuals, there's perhaps even more striking heterogeneity in the layout and orchestration of functional brain networks. So in these examples from the Midnight Scan Club dataset, you can see first on the left-hand side that each functionally defined region tends to be different in its shape, size, or location across individuals, with some regions being lost entirely in the group average, as shown by the black arrows. In the plot on the right-hand side, white outlines depict a functionally defined region overlaid on top of resting state data from one individual. And you can see that although these match up pretty well in the top left subplot within a single individual, they don't match up at all across individuals as shown in the other panels. So for these reasons, it's clear how important it is for us to develop strong methods for doing personalized neuroscience, capturing each individual's unique variability in cognition, clinical phenotypes, and functional brain organization. However, this approach does come with some unique challenges. Specifically, we seek to balance the individual granularity that comes with doing personalized neuroscience with things like reproducibility, generalizability, interpretability, and actionability, all of which are made more and more difficult when our neuroscience approaches get more and more personalized. So to achieve a balance between these two, I will argue that we need machine learning methods that have the right types of constraints. So constraints that help us avoid overfitting and yield actionable interpretations of results that are true across individuals. So today, I'll show two examples of how we might accomplish this, both in developmental datasets. First, I'll talk about a project in which we attempt to predict individual differences in youth cognition with personalized functional brain networks. Next, I'll discuss how we build predictive models of borderline personality symptoms using functional connectivity. And then I'll wrap up with a summary and some future directions. So first, how might we derive personalized functional brain networks? The classic approach that I'm sure many of you are already familiar with is to take a single atlas of functional brain networks derived by group averaging and warp it to fit the shape and size of each individual brain, making the assumption that structure and function have a one-to-one mapping. This approach falls short when this assumption is not met, particularly where functional brain networks show inter-individual heterogeneity. This is especially problematic for association networks such as the fronto-parietal network that support higher order cognition, since these networks tend to be the ones that have the highest degree of variability. The approach that we've taken to deriving personalized functional brain networks uses non-negative matrix factorization. So we begin with a matrix of activity at each vertex over time as we record from the scanner, and we decompose that matrix into a matrix of network loadings by vertices and a matrix of network activity over time. This procedure will positively weight covarying elements, allowing us to extract functionally connected systems within each individual. Importantly, we include two key constraints. First, a group consensus regularization term that preserves inter-individual correspondence, making it easier to interpret results across individuals. In essence, this means that we can map each subject's individual set of networks back to a recognizable set across individuals. And second, we include a data locality regularization term, which improves the robustness to imaging noise and improves spatial smoothness, essentially helping us to avoid overfitting. So here's the result of our 17 network solution, wherein each network has been named according to its alignment with the Yo atlas. This solution was first derived in a sample of youth from the Philadelphia Neurodevelopmental cohort and in a previously published paper by a former member of our lab, Zai Xu, in 2020. So today I'm going to show you results from a replication and extension that I conducted of this study in the ABCD dataset, which is much larger. So the Adolescent Brain and Cognitive Development Study recruited nearly 12,000 individuals aged 9 to 10 years old and has been following their development over time. So today I'll show you the results from the baseline cohorts, neuroimaging and cognitive measures, though we are excited to follow this work up with longitudinal analyses as more and more data is released. So we excluded participants who had excessive head motion, images that didn't pass QC, or incomplete data, yielding a sample of around 7,000 kids. We then split this sample into discovery and replication subsamples, each with about 3,500 participants. And we used a previously published group assignment for this matching procedure, which ensured that we had matching across a number of different variables, some of which I've listed here, but they include things like age, sex, and a number of socio-demographic variables. To assess cognition across multiple domains, we used 3 domains derived from a previously published study in the same dataset by Wes Thompson and colleagues, where they applied a Bayesian probabilistic principal components analysis across a battery of cognitive tasks. And these 3 domains capture the majority of variance across the cognitive tasks, with the tasks that are loading highest on each domain listed below each one. To investigate associations between PFN topography and cognition, we applied linear mixed effects models. So we calculated the total cortical representation of each PFN by summing the loadings for that network across each vertex. In each linear mixed effects model, we included age, sex, motion, and family ID as covariates, and we applied combat harmonization to harmonize the data across collection sites. First, we observed that association networks, such as the fronto-parietal network shown in yellow, were more strongly associated with cognition than somatomotor networks. And this was true across both studies, including both the discovery and replication subsamples in ABCD. Replicating these results further, we see that the fronto-parietal networks 15 and 17 are significantly associated with cognition across all 3 samples, though we observe smaller effect sizes in the much larger ABCD dataset, as we may expect. Next, we aim to build predictive models of individual differences in cognition using PFN topography. So to do so, we applied ridge regression, using the loadings of each network at each vertex to predict individual differences in cognition. The ridge constraint here is important because it shrinks the weights on each parameter towards 0 to help avoid overfitting to the training data. And then we use cross-validation, in this case, tenfold cross-validation, to assess how well we could predict cognitive abilities in unseen participants' data. We again covariate for age, sex, and motion, harmonized the data across sites, and made sure that siblings stayed within the same folds to help avoid biasing our results. On the left are the results from the original study, and on the right is our replication in ABCD. In both cases, showing a significant correlation between the actual cognitive performance on the x-axis and the cognitive performance predicted by the model on the y-axis. Results are shown here for the 2 matched samples in black and gray, with the inset histograms showing null distributions generated by randomly scrambling that outcome variable. Across both datasets, we also see that the same personalized functional brain networks are contributing to the predictive accuracy, with a bias toward association networks such as the fronto-parietal and ventral attention networks. Having replicated these key results from this previous study in a much larger sample of youth with remarkably similar results, we next sought to extend these findings in a couple of key ways. First, we explored whether we could get good predictions for performance in other cognitive domains from these same networks. So here I'm showing the prediction accuracy results across general cognition, executive function, and learning and memory. And we were able to achieve pretty good predictions for the other 2 domains, though not as strong as for general cognition, which most closely aligned conceptually with the EF domain from the previous study. Interestingly, we found somewhat similar results across the 3 domains in terms of which networks were contributing most to the prediction accuracy, with the association networks again dominating and having the greatest contributions. This motivated us to explore whether there was a relationship between the prediction accuracy and a given network's position across a principal axis of cortical hierarchy. So the sensory motor association axis, or SA axis, describes a dominant hierarchical organization pattern in functional systems across the brain. Specifically, regions are ranked according to their position along this hierarchy, from the sensory motor pole, shown in yellow, to the association pole, shown in purple. This map is actually derived by averaging across multiple other maps, showing remarkably similar patterns across the cortex, including functional, anatomical, hierarchical, and molecular gradients. Looking at our brain maps showing the networks that are most strongly predictive of cognition, we wondered whether a given network's average rank along the SA axis was associated with its prediction accuracy. And indeed, across all three domains of cognition, we find that regions ranked higher on the SA axis have functional topography that's more predictive of individual differences in cognitive performance. So to summarize this first section, we conducted a successful replication of a previously published study showing that personalized functional brain networks are associated with and can predict individual differences in cognition. We then extended these results to include 3 domains of cognition and found that hierarchical position along the SA axis is positively associated with prediction accuracy. Next, I'll talk about our project in which we attempted to predict borderline personality symptoms with functional connectivity. So borderline personality disorder is defined as a pervasive pattern of instability of interpersonal relationships, self-image, and affects, and is marked by impulsivity beginning in early childhood and present in a variety of contexts. Unfortunately, previous imaging studies of BPD have typically been very small. And as we know, small samples tend to produce inflated results. So in this study, led by Max Bertolero from our lab, we aimed to conduct the largest imaging study of functional networks related to borderline symptoms. Specifically, we sought to derive a reproducible functional brain network signature of borderline symptoms by discovering and replicating results in independent samples. And we investigated associations with the development of functional brain networks, and importantly, aimed to conduct this study using an open science approach, sharing all of our data and code. To assess borderline personality spectrum symptoms, we used the NEO 5-factor inventory, a widely used instrument that's been validated across 6 datasets from both clinic and community samples, and it's highly correlated with borderline-specific scales as well. So the data sets that we used for this study are the Human Connectome Project Young Adult Sample, or HCP-YA, and the Human Connectome Project Development Sample, or HCP-D. In HCP-D, only participants who were at least 16 years old had the borderline symptom assessment. So in total, we had a sample of about 1,100 individuals across both studies. Our analysis steps were as follows. First, we took as input the functional connectivity matrices derived from the HCP-YA sample. We then modeled the relationship between functional connectivity and BPS symptoms for each of 400 regions. Next, we tested the prediction of these models for each region in the HCP-D sample, applying the pre-trained model from HCP-YA for each subject. After assessing how well these models trained on HCP-YA data could predict BPS symptoms in HCP-D data, we then compared the brain maps of prediction accuracy across regions to brain maps highlighting regions undergoing changes during development. We found that we were indeed able to predict borderline personality symptoms in young adults from HCP-YA using cross-validation. And we were able to successfully apply these models trained on HCP-YA data to unseen HCP-D data without any additional tuning. So next, we compared brain maps showing which regions contributed most to that prediction accuracy in HCP-YA to maps showing the regions of most developmental change in HCP-D. So looking at the overlap between these 2 brain maps, we find that developmental and borderline symptom features overlap in the TPJ, retrosplenial cortex, and anterior insula, highlighting these regions as potentially useful biomarkers to investigate in future studies. To confirm that this overlap did not occur by chance, we compared the observed alignment with the null distribution that was generated using a conservative spin-based permutation testing procedure and found that the true alignment, shown in the black line, was significantly more overlapping than chance. So to summarize, we conducted the largest study of functional networks linked to borderline personality spectrum symptoms. We found robust results when our models were applied to unseen data in both adults and older adolescents. And finally, we observed that functional connectivity patterns that were predictive of BPS symptoms were also those that underwent the most development in youth. So to summarize, returning to our problem of balancing personalized neuroscience approaches with reproducibility, generalizability, interpretability, and actionability, I've shown 2 examples of studies in which machine learning algorithms with appropriate constraints can help us achieve this critical balance. We've made use of both supervised and unsupervised models to predict individual differences in both cognition and psychiatric symptoms, with predictions that are readily interpretable and generalize well across multiple developmental data sets. In our ongoing work and future directions, we're interested in further understanding how different types of experiences and environments shape this neurocognitive development, again, by balancing between personalized approaches and the need for generalizability. We're currently investigating associations between personalized functional brain network topography and different aspects of an individual's environment and exposures. We're also interested in replicating and extending our results with borderline personality disorder, looking to apply these models in a 3rd data set and expand our models to include subcortical regions as well. So with that, I'd like to thank my mentor, Ted Satterthwaite, as well as Max Bertolero, who led the study on borderline personality, and the rest of our wonderful lab mates and extraordinary team of collaborators as well. And I'd be happy to take any questions, so please reach out. Thank you.